Hi, this is Christian and Daniel's meiosis Explain Everything project. Meiosis is a type of cell division that creates female egg cells or male sperm cells, and this happens through meiosis 1 and meiosis 2. However, before any of the division can occur, the chromosomes must replicate during interphase, and the purpose of this is so that by the end of meiosis 2, the daughter cells formed will have half as many chromosomes as the original cell, so when it fuses with another gamete, the unified cell will be diploid. And a diploid cell is a cell that has a full set of chromosomes. After all of the replication in interphase 1, the homologous pairs will cross over, and this is when a part of the chromatid breaks off and then rejoins with the other chromatid. And as you can see around this area where the two junctions meet, that is known as the chiasma. The purpose of the crossing over is to promote genetic variation. This happens when the crossed over genes are reshuffled so that it could produce a new combination such as capital C, lowercase d, and lowercase c and uppercase d. Also, it increases the number of allele combinations to the point that the number is close to infinity. As you may be wondering, an allele is one of the possible forms of a gene, and crossing over of non-sister chromatids produces new combination of alleles because when the junctions break and the non-sister chromatid reconnects with the other sister chromatid, different alleles are matched together, and this creates different combinations and genetic variety. And then in metaphase 1, the homologous pairs meet within the middle of the cell, and then spindle fibers start attaching to the centromeres. And homologous pairs are chromosomes that contain a pair that comes from the mother and father, and this allows them to carry the same gene, but the alleles for the traits can be different. After the spindle fibers have attached to the centromeres of the homologous pairs, the spindle fibers will start retracting towards the centriole, and this will cause the homologous pairs to be pulled apart and to opposite ends of the cell. However, some people may be wondering if the splitting of the homologous pairs is random, and I can assure them that it is, because in meiosis, each chromosome is attached to only one pole, not both. Also, the orientation of the homologous pairs is random, so that each chromosome has an equal chance of connecting with a spindle fiber and splitting apart. Lastly, the orientation of a homologous pair will not affect another homologous pair. So this will allow equal chances for each pair to connect the spindle fiber with its centriole no matter how it is faced.
Also known as Mendel's second law, Mendel's law of independent assortment states that the alleles separate independently during the formation of gametes. This means that traits are transmitted to offspring independently of one another. In meiosis 1, members of homologous pairs separate during anaphase. The separation of each pair is random with regards to all the other pairs. Each pair consists of a maternal and a parental chromosome which originated from the mother and father. This will ultimately lead to offsprings being more genetically variable than if the parent chromosomes were always associated with each other.
While the homologous pairs are being pulled to opposite ends of the cell, one chromosome from each pair is actually pulled to each side, and this disjunction is the main cause for the number of chromosomes to be halved. Also, when they are pulled to the opposite side of the cell, a nuclear membrane forms around the chromosomes, and the cell splits through cytokinesis, and this is what transforms the cell from being diploid to haploid. Also, as you can see in the haploid daughter cells, the two alleles of each genes are separated to the respective nucleus, as you can tell by the red and black and red.
In prophase 2, the nuclear membrane and nuclei break up, while the spindle network appears and the chromosomes, which consists of 2 chromatids, condense and become visible. These 2 chromatids are called sister chromatids, which are identical copies of each other, and they are formed during replication that are joined by a centromere. As you can see from the slide, the chromosomes align in the metaphase plate in preparation for centromeres to divide in the next phase. Anaphase 2 is very similar to anaphase 1. The sister chromatids separate and move towards the opposite cell poles. In telophase 2, distinct nuclei form at the opposite poles and cytokinesis occurs, which ends up splitting the cells into 4 daughter cells, each with 1/2 the number of chromosomes of the original parent cell. Since chromosomes undergo crossing over in prophase and are pulled apart in different combinations during metaphase and telophase, gametes containing different combinations of chromosomes will unify to form a zygote. Each zygote will have a different combination of chromosomes. These different combinations will be creating variants among the genes because each parent can offer different types of chromosomes. More specifically, each parent is providing half of the total pairs of chromosomes needed, so when the male and female gamete unify, the zygote will acquire a full set of chromosomes and become diploid. Also, when the gametes unify, one of the parent chromosomes containing an allele will bind with the other parent chromosomes containing another allele. This random unification of the gametes can result in homologous or heterozygous allele pairs, which can express different genes. Also, as you can see from the slide, the gametes contain only one allele.